I'm so honored to be here, and I want to thank the steering committee. I want to thank Lorraine and Emigdio and Louis and Vicky, and thank the people of this land. This may be one of the smallest audiences that I've spoken in front of, but this is probably the most significant, and I say that with great sincerity. A recent quote from a Native American elder, and I wish I knew the name, so if anyone knows who spoke these words, I want to give proper credit. But he said that we do not inherit our environment from our ancestors. We borrow it from our descendants. And I think that is so true. In the next hour and 20 minutes, I'm going to show you some things that I think are self-evident truths. And nature is unbelievable in this complexity of design and successes and creations. I am, in a sense, rediscovering that which our elders long ago discovered, that mushrooms are deep reservoirs of very important medicines. And I stand here, and I wear my favorite hat. This is my hat. It makes me look rather dapper, and I'm balding on top, so it keeps me warm. And it is made from this mushroom, which is called amadou. The Latin binomial is called Fomes fomentarius. Amadou is one of the most important entities that humans associated with for human survival. Indeed, it can be said that a lot of us here today are because of this mushroom. There's no doubt more than 100,000 years ago we migrated from Africa. We traveled north. We discovered something new in Europe called winter. Oops. This mushroom allowed for the portability of fire. You can hollow this mushroom out, put embers of fire inside, and carry fire for days. And as this audience knows in particular, the firekeeper of the clan may now hold a ceremonial role, but not too long ago, the role was extremely important for the survival of the clan. When this mushroom is boiled in water, it delaminates into mycelium, and you'll see lots of mycelium. And some ladies in Transylvania made this hat, and so this tradition goes back thousands of years into Europe. Now, this mushroom was first described by Hippocrates in 450 BC. It is the oldest known documented medicinal mushroom. No doubt, our ancestors were using these mushrooms for thousands of years prior to that. And I brought another fungal friend with me. And this is agarikon, the longest living mushroom in the world. It's resident in the old-growth forest of British Columbia, Washington, Oregon, and Northern California. It perilously survives in sky islands in Austria and Slovenia and northern Italy. Because the old-growth forests have been cut down, this species is now on the red list of extinction in Europe. This mushroom was described by Dioscorides in 65 AD as elixirium ad longum vitam, the elixir of long life. And I suggest to you today that this mushroom will be as important for human survival as amadou proved to be in the past. So I'm going to take you on a mycological mystery tour, which I'm really excited to show you because it's the culmination of not only my lifetime, but then that of many others who preceded me. This is the great earth that we live upon, and I believe that following into the future, if we follow the path of mycelium, we will lead to greater sustainability and greater biodiversity. I had an epiphany, and I like one of the waking moments. I like to lay in bed and just ponder the day. Just take 5 or 10 extra minutes and just think through the important issues that you need to consider. And this epiphany came to me out of the ether that habitats of humans share immune systems, and mushroom mycelia are the cellular bridges between the two. I sincerely believe this is true. I'd like to give respect to my teachers. These are my mentors, Dr. Alexander Smith, Dr. Daniel Stuntz, Kit Skates. These 3 individuals have now passed on, and Dr. Michael Bugh, who is my professor at the Evergreen State College. So rare is agarikon that in 40 years of going into the old-growth forest, Mike found his first agarikon this past fall. And this is group of individuals, with the exception of Mike, but these individuals in today's political context you would consider to be conservatives, political conservatives. I met them when I was about 19 years of age, and they adopted me as their sort of mascot, their student. All the more surprising because when I was 19, this is what I looked like. Your suspicions are now confirmed, okay? So I'm going to progress through the historical record on the use of mushrooms. This is from northern Algeria, from the TASCL Plateau, which was called the Plateau of Running Rivers. Now, because of desertification, the Sahara Desert has encroached upon that environment. But about 5 of these pictographs were found in these labyrinthine cave complex in northern Algeria. And here's an example of what we call mycophobia, the irrational fear of fungi, which has actually permeated the scientific community as well. Even though there's 4 different peer-reviewed articles on the art of the TASCL Plateau that featured this image here, which has been redrawn by my friend Jonathan Meter, not a single scientist dared to suggest even, hypothetically, what the artist intended to describe. Obviously, the artist is very excited about mushrooms. And the association of the bee god and bee with mushrooms was an ancient method of preserving these mushrooms. And maybe it's not surprising that there is mycophobia, the fear of fungi, because in your encounter with plants and animals, your experiential window of that encounter expands weeks, months, years. Mushrooms are ephemeral, most of them. They come up just for a few days and they disappear. And to find mushrooms that are so short-lived in your viewscape of your life, that can feed you, that can kill you, that can send you on a spiritual journey, no wonder that we've lagged behind in our understanding of these mushrooms. But our ancestors knew a lot more, perhaps, about mushrooms than we do today. Another image 7,000 years ago in the cave, clearly this artist is communicating that the mushrooms are giving something to the mind. And I think this transcends time. And then this was discovered in Spain. This is a group of mushrooms that are associated with oxen. And this group of mushrooms here is thought to be putatively Psilocybe hispanica, a magic mushroom. An article was just published in the Journal of Ethnobotany specifically on this mushroom. And when we advance forward to about 300 to 400 years BC, this speaks to the origination myth of women, of the seasons in Greek culture, where Demeter is giving Persephone a mushroom, and as Persephone consumes the mushroom, she's taken into the underworld. That's the beginning of winter, and then renewal upon spring, upon her coming from the underworld up to the earth, and then the spring and the seasons were reborn. This is the division between Austria and Italy. And in 1991, a group of tourists discovered a body frozen in the ice. And because the glaciers were thawing, because of climate change, the body was uncovered. And these tourists got really excited. They found this frozen individual who obviously was very, very ancient. And they contacted, this is the border, and they contacted the Italian authorities, the border guards. And the border guards literally said, oh, another dead hiker, we'll come see him later. And these tourists realized, no, this body's thawing out, this is really important. So they went on to the Austrian side of the border, and they informed the Austrian officials, who came over and saw the body, and they said virtually, the father of the Austrian people, we found you. And they dragged him across the border, which created an international incident, which has now he's been repatriated back to Italy. But this is Otzi, which is the best preserved remnants of a human that's ever been found in a non-funeral setting. So he died basically as he had a spear point in his back, and tethered to his right side with 2 of these polypore mushrooms, one of which was Piptoporus specialinus, a birch polypore, like this mushroom, and the other one was the same species. And here it is here. And this fabric that was tethered to his right side was a fire starter fabric that is made from this mushroom. Now, on our right side, for those of us who are right-handed, I have my knife, I have in this case, my laser pointer, and my car keys. We keep the items of the most important, of greatest importance, close at hand. And this also this position with the Iceman tethered to his right side was deemed to be significant because it's so important that he could keep fire and start fire, and he needed his fire starting toolkit to be very easily accessible. He needed to know where it was so he would not lose it, because to lose it could mean death. So I customized this presentation for this group here, but I thought this is important to bring out. This is some Anasazi petroglyphs that are extraordinarily unusual. And here are clearly, from my taxonomic viewpoint, these are mushrooms. When our peoples migrated over the land bridge in Alaska and came down, well, we had the knowledge of the resident organisms in our ecosystem. But then as the ecosystem became more arid, then we would lose many of these species. So this petroglyph has not been dated, but I think it must go back many, many thousands of years when these mushrooms were also resident within this ecosystem. And the Prince of Flowers, Cocopili in Mexico City, is adorned with many of the sacred plants. These are mushrooms. The incurled margin here is significant because this speaks specifically of a group of mushrooms which are magic mushrooms called the psilocybin species, or otherwise known as psilocybes. This is Psilocybe hopeii, a new species that has been discovered in the San Francisco mountain range north of Flagstaff, Arizona. And it is a species that's been named in honor of the Hopi people. So these mushrooms do and have grown in this area. And they're probably resident now in little islands, shrinking islands of ecosystems where the remnants of these species are likely to still exist. My wife and I spend a lot of time in the old growth forest. This is where we go to church on Sundays. And the old growth forest has become our large laboratory of experience. The old growth forest is something that I have a passionate interest and affection for. It draws me in, and it's a place that I use for my meditations. But when we look at an ecosystem, we now know something very, very different than science knew before, that all plants are part fungi. In Science, which is a journal, there was actually an article published a few years ago saying that all plants are part fungi. And so when you look at the cross-section of an ecosystem, there are the parasitic fungi that kill the trees, and then after the trees are killed, there can be saprophytic fungi that then rot the trees. And then there are the endophytic fungi, which are extraordinarily interesting. Several hundred endophytic fungi are growing parallel to and inside the tissue of plants. Scientists were perplexed when they do tissue culture in the laboratory of plants. They kept on getting fungi that thought they were contaminants. We now know that these are endophytic fungi. They're mutualists. They help the plants survive. They create a host defensive resistance against pathogens. And that plurality and complexity of the fungal population existing within the plants and on the plants gives the plants to be able to resistance against disease and drought. And then the mycorrhizal fungi, an extraordinary group of species which can extend the root zones hundreds of times. The mycorrhizal fungi get sugar from the roots and the up-channel minerals. Here is a root that's coming near to the surface. As the mycelium reaches the surface, it exhales carbon dioxide, inhales oxygen. In fact, fungi respond to 4 primary environmental stimuli. When there's an influx of water, well, we all know that. Mushrooms like water. But coincident with that, there's usually a drop of temperature as evaporation occurs and the clouds block the sun. So an influx of water, a drop of temperature, the mycelium comes to the surface, exhales oxygen, exhales carbon dioxide, inhales oxygen, and then they're exposed to light. The majority of mushrooms are photosensitive. They require light stimulation for them to form. So looking at the mycorrhizal fungi, they're spectacular in their ability, you can demonstrate the usefulness, something I very much encourage the community here to experiment with the mycorrhizal fungi. These are poppy seeds that were planted first into soil that did not have mycorrhizae, and the seeds here were dusted with mycorrhizal spores and planted side by side. One of my employees is a big skeptic. He did this in his backyard going, I don't believe this. And then he did this experiment on his own, and he's become a true believer. And an example also is all trees have these mycorrhizal fungi that also aid their growth. So when we look into the old growth forest, and we look at these hemlock trees that grow in nurse logs, scientists wondered how is it possible these trees could have enough light to be able to photosynthesize. When they took these hemlock trees and they put them into a greenhouse environment, gave the same amount of light that they were getting here in the old growth forest, all the trees died. They wondered how is it possible those trees could survive in the old growth forest. Two scientists get credit, Arnbrandt, 1994, Simard, 1996. When they radioactively tagged carbon and nitrogen around the surrounding environment, they found something heretofore unknown in the field of science, that in the Populaceae, including aspens, cottonwoods, and alder trees, in this case, alder trees and aspens, this has been shown, will move nutrients hundreds of feet in the sunlit riparian zones where these trees typically grow, and move it hundreds of feet to feed and nurture these small conifer trees. For the first time, it was known that mushroom mycelium bidirectionally budgets nutrients to many plants, including trees in the forest ecosystem, to support the biodiversity and the plurality of species within the ecosystem, because that biodiversity gave the ecosystem resiliency to catastrophia, to sudden changes. And so for the first time, we understood that the mycelium had a long-term view of the ecosystem to allocate resources so that complexity can be preserved. This is the largest mushroom in North America. It's called Brygioporus nobilismus. It weighs several hundred pounds. We've tried cultivating it many, many times. Prodigious producer of white spores. It weighs several hundred pounds. This species also grows exclusively in the old growth forest. So the mycelium, when it grows, becomes tenacious. It holds soils together. The fungi are the grand molecular disassemblers of nature. They decompose plant and animal tissue, and in doing so, they build soil. So they are the soil magicians. But in the course of their growth, they produce these wonderful root-like structures called mycelium. And this form of mycelium is called a rhizomorph, a root form, because it's so tenaciously can hold the soil together. I did a measurement with a Mettler balance. Some of you will know what that is, a very sensitive balance. And I got a 30,000 times to 1. When I put 1 centimeter, weighed this 1 centimeter long, and then I weighed this, the ratio of the mass was 30,000 to 1. In fact, I could have made it 1/10 that long, a millimeter in length. You know, it would have been 300,000 times to 1. So when the mycelium goes through the habitat, it holds it together, it grips it, it prevents erosion, but it does so much more than that. This is my manly man photograph. For 3 years, I was a logger. I set chokers in the Olympics. I was a long-haired hippie, as you saw at the time. I got enormous amounts of grief. The logging community can be extremely cruel, especially to new young men. And I survived, and not only I survived, but I was elevated to a key position. Then 3 of my teammates were killed in a logging accident. I was running Skylines. They wanted to elevate me at the age of 22 to be the leader of the entire crew. And I decided I'm going back to college. But looking at these trees when they're down, they do so much more than what many people had thought before. But in essence, the saprophytic fungi take wood. The wood becomes colonized with mycelium. Lots of things happen. Ultimately, soils being generated. Fungi create soils. And this is a soil horizon, not too far from where we live. Here's the clay layer. And this soil has been developed over 10,000 years. We live on an extremely thin film of soil. And the erosion of that soil causing the loss of biodiversity and is pulling apart the very food webs that would otherwise give us life. So these fungi play a critically important role for creating the very life support systems that are part of the food web. The mycelium then is triggered into mushroom formation. Again, a drop in temperature, influx of water, exhaling carbon dioxide, inhaling oxygen, and then exposure to light. Suddenly, the mycelium then concentrates into a little mushroom, and the mushroom then expands within a matter of 2 or 3 days. Mushrooms do not have a good immune system. The mycelium does. The mushrooms will rot, and they're only up for 4 or 5 days at most for most of these species. But like ripened fruit, you know, they attract insects. And the ripened fruit then is quickly exploited by other members in the ecological community. Mushrooms, upon sporulation, upon giving off their offspring, then quickly rot. And the organisms that rot the mushrooms we believe, or I believe, is absolutely critical for the downstream environments and setting the stage for rebounding the ecosystem that originally gave it life. So spores are being produced underneath the gills. This is a portobello species. Here's oyster mushrooms day 21, day 23, day 25. You can get 5 pounds of oyster mushrooms, wet weight, from 5 pounds of dry sawdust or straw. And then the mycelium is edible for the cows, chickens, and pigs. And it does so much more than that. But upon sporulation, these mushrooms are extremely perishable. And so as they begin to rot in the old growth forest here, and then they sporulate, lots of other things are happening, and then the mycelium goes subterranean. This is the majority of the life cycle of mushrooms, is at the mycelial state. It's like the tip of the proverbial biological or mycological iceberg. The mycelium is resident in the ecosystem for hundreds of years, and mushrooms only come up seasonally, and sometimes decades in between the times that you'll see them. In a single cubic inch of soil, there can be more than 8 miles of these cells. My foot covers about 300 miles of mycelium. Think of that. 300 miles of mycelium if you stretch these cells end upon end. I spent many years as a scanning electron microscopist. This is one of my scanning electron micrographs. And the mycelium, when it produces this web, is beautifully articulated. It's a microfiltration membrane, so there's a flow of nutrients, like the rains that we just had yesterday, then is captured in this net, and the mycelium externally digests its nutrients. We know now from DNA analysis that we are more closely related to fungi than we are to any other kingdom. 650 million years ago, we split from fungi. In the Journal of Eukaryotic Microbiology, 4 years ago, 23 scientists published a new super kingdom called Apisthoconta, joining Animalia and fungi together, because we're more closely related to fungi than we are to any other kingdom, and our cells under the microscope look almost identical. We exhale carbon dioxide, we inhale oxygen, so do fungi. But when we look at this network, you know, these little microcavities swell with water, so myceliated habitats hold water together. This is like mulch, what's full of mycelium can hold water together. And then as the rains go away and the dry season occurs, then these little cavities release water one at a time, like little bladders. And so you have sustained moisture over the long term because of the microcavities. And moreover, the pre-selection of the bacterial communities that grow within these cavities is determined by the mushroom species that is permeating throughout the ecosystem. So they set the stages for microbial successionism that leads to the plants that create the debris fields that then the fungi can feed upon. These are 2 movies I'm going to show you by my friend Patrick Hickey. First, there's territorial conquest. The mycelium grows out, and then it consolidates its control. This next movie has revolutionized our understanding of what's happening microscopically within the mycelial networks. The fungi produce bundles of nuclei called comet nuclei, and these hundreds of nuclei streams to the tips. And so the cells are normally binucleate. They have 2 nuclei per cell. But as they actively grow throughout an ecosystem, they become hypernucleate. And in the meter diameter patch of mycelium, there can be literally trillions and trillions of end branchings. And if there's a new insect, a new food, a new toxic waste that's never encountered in the evolution of these fungi, just think of these, the end branchings being scientific experiments. If there's trillions and trillions of experiments, and one experiment is successful, and it generates a new enzyme, a new acid, a new strategy for gobbling up that potential food source, what happens? The mycelium streams. But some of that information via the nuclei back channels into the mother mycelium. The whole mat becomes educated. This is the concept and the foundation of epigenesis, which is a big buzzword now in biology, is the expression genetically of an organism in response to environmental stimuli. We have a lot of genetic expressions which are not expressed until we are challenged in nature. And so this has now made us much more clearly focused on the role of mycelium in nature and the fact that its network-like design allows it to create experiments that lead to the ability to adapt. So I postulated in the early 1990s that fungi were intelligent. I got a lot of accusations of people saying I was eating too many magic mushrooms, which I do not deny. But this group of scientists in Japan put the slime mold, Physarum polycephalum, into a maze, and the slime mold grew out randomly. They put 2 oat flakes at 5 exit points, 2 out of 5. Then the slime mold would grow through the maze, and then it would shut down the non-essential pathways and grew directly to the sources of nutrition, disregarding any dead ends. Thus, they postulated this is a form of cellular intelligence. I would agree. But then the Japanese love doing things like this. And here's another Japanese group, more recently, in 2010, decided, well, if the slime molds are intelligent, how would they redesign the Japanese subway system? And so here is Tokyo, and here are the different satellite cities around Tokyo. They put the slime mold in the center, these all nutritional flakes that represent the sub-cities around Tokyo. They inoculated the slime mold, and it grew out, and you can see it goes out and makes connection to these food sources, and there's lots of other random branchings. In 26 hours, it shut down all the non-essential branches and redesigned the Japanese subway system in a more efficient manner than it is designed today. Moreover, when they applied mathematical analysis, the slime mold created a mathematical optimum. There is no better way of solving this problem than that which the slime mold had achieved. So if you have an engineering issue, you might want to consider consulting a slime mold. So I'm going to wax poetic here, but this is central to my belief system, and I don't think it's going to be strange to most people in this audience. The mycelium conforms to an archetype. That's similar to our brain neurons. This is a photomicrograph by Hank Morgan. And you look at the design of the mycelial network, it dawned on me that these looked like neurons, and the mycelial network conforms to the same archetype upon which our brains are based. And then looking at the computer internet, I was astonished when I saw this diagrammatic representation of the computer internet also conforms to the same architecture and the archetype of that of the mycelium. I believe the invention of the computer internet is an inevitable consequence of a previously proven evolutionary successful model. The Earth was bound to invent the computer internet at a time critical when information and resources need to be shared in order to prevent extinction. And so going further out, we're looking at the formation of dark matter. Most of you know that 95% of the matter in the universe cannot be seen. And the structure of dark matter also conforms to the mycelial archetype. And from Scientific American, the Deep Hubble Telescope view, this is 170,000 galaxies in this viewscape. These are the galaxies, and the cobweb of dark matter also conforms to the same archetype. I believe matter begets life. Life becomes single cells become strings, strings branch to become membranes, and this is the way. We will find membrane, mycelial-like organisms throughout the cosmos. So let's go back in time. The first organisms that come to land were fungi more than 1 million years ago. Plants followed several hundred million years later. Fungi came to land first and associated with algae to form lichens. Most of you know that lichens are a mutualistic relationship between algae that can grow separately and fungi that can grow separately, but when they come together, they form lichens. And then the mycorrhizal fungi, the plants teamed up with the fungi to help them be able to march onto land. Well, the fungi are producing exotic chemicals in these extracellular droplets, and one group are oxalic acids, which are 2 carbon dioxide molecules joined together. Very interesting. And then they mineralize rocks. That's the term of art that's used. They actually pull minerals out of rocks. So they pull out calcium, iron, manganese, and this mineralization of rocks caused the rocks literally to be pockmarked. They have cavities. And so when it rains, the cavities fill up with water, so the rocks are no longer smooth. It leads to the crumbling of rocks and the formation of calcium oxalates. And the calcium oxalates are insoluble salts. So once these salts are formed, they sequester carbon dioxide. It's counterintuitive because fungi produce CO2, but they produce enormous amounts of carbon that's sequestered in the environment. And these salts then become resident in the ecosystem until enzyme or acids of another organism redissolves them. Working with Battelle Laboratories, we looked at these calcium oxalates And our hypothesis is the calcium oxalates go off and advancing on an osmotic pressure wave in advance of the mycelium. And as they encounter a bacterium, for instance, as the enzymes of the bacterium cause these calcium oxalates to degenerate, the chemical scent trails then are picked up with the mycelium, which then tells the mycelium what is in advance of contact. And so the mycelium then through epigenesis is able to create a response mechanism that will make it more successful in combating potential adversaries. And I'm such an idiot. It took me forever to realize this. I would be tipping over rocks all the time and seeing mycelium. I never knew for the longest time that the mycelium actually was munching the rocks. The mycelium eat the rocks. And here's a clear example, the mycelium marching up into the rocks, pulling out these minerals and then channeling it to nearby plants. So this is how plants get the majority of their minerals, is through these mycorrhizal fungi. This fossil was discovered in 1859, laying down, and it grew between 420 to 380 million years ago. Now, at that time, 420 to 380 million years ago, that's before vascular plants, before flying insects. And so there were ferns, but there weren't trees, basically. And lying down 3 feet tall, 420 million years ago, this was the tallest organism on land on this planet. Well, it was a big mystery what this organism was. It was given the name Prototaxides. And for over 150 years, as a matter of scientific debate, until Dr. Kevin Boyce, a few years ago, in the Journal of Geology, published a series of articles and republished articles in response to the challengers. But we know now, de facto, proven by very good scientific method, that 420 million years ago, dotting across the face of the earth, were these giant fungi, over 30 feet in height. And they would attract lightning, of course. You know, lots of things would happen because of that. But think of that. Dotting across the earth were these giant mushrooms, 30 feet tall, before vascular plants, before flying insects. They would be a great reservoir, a great vessel for all sorts of other organisms to live within. So as we advance now forward from 380 million years ago to 250 million years ago, it was a time when all the continents were combined, at the time of Pangea. 250 million years ago, there was a great extinction event. More than 90% of the species in the fossil record, you can clearly see it, became extinct. And the extinction event was caused by potentially 3 different events. And it's a matter of scientific debate still. There could have been an asteroid impact that jettisoned enormous amounts of debris in the fossil record. There's an iridium layer that can be found all over the world at one specific location 250 million years ago. There could have been volcanoes in Eurasia, or there could have been methane hydrate bursts from the ocean. I don't see these as being mutually exclusive. The asteroid impact could have created the earthquakes that caused the volcanoes to explode, that caused the methane hydrate to burst, but we know one thing for a fact. The extinction occurred, sunlight was choked out, and fungi inherited the earth. Those organisms that paired with fungi had a better chance of survival. We actually have the name of this fungus. It's called Reduviosporinites. That is evident in the fossil record, now extinct today, but this is the fungus that gobbled up the forests that quickly died. Then we advanced to the time of Gondwanaland, 140 million years ago, when we have continental drift. And then 65 million years ago, we have another huge extinction event. This time we know what it is. It's not a matter of scientific dispute. An asteroid impact occurred. And 65 million years ago, it hit directly above where the Yucatan is. Enormous amounts of debris jettisoned into the atmosphere. We can see this again in the fossil record. Massive extinction. Sunlight is choked off, and fungi re-inherited the earth. There's a lesson here. As we are exposed to extinction events, those organisms that pair with fungi tend to survive. We've now entered into 6X, the 6th greatest extinction event known in the history of life on this planet. Dr. E.O. Wilson from Harvard estimates that more than 50% of the known species will become extinct in the next 100 years. We are probably losing species faster than we can identify them. And it's so important that we understand that for the first time, this extinction event is being caused by an organism, by us. We are not only the cause of this extinction event, but we're likely to be its victims. So I'm going to zoom down to, we live here in Washington State. Here's the Columbia River. And here is the largest organism in the world. And it is a honey mushroom that kills the trees. Armillaria astoi. It's an edible and choice mushroom, by the way. Kills the trees, but then the Forest Service wants to cut the trees down because of fire hazard. And we knew the GPS coordinates of this fungus that was reported in the scientific literature. It is 2,200 acres in size, 1,665 football fields, and it's one cell wall thick. We have 5 or 6 skin cells that protect us from infection. The mycelium has 1. If you've ever done soil analysis, many of you have, it's not uncommon to find several 100 million organisms per gram of soil. That's a testimonial to healthy soil. And think the largest organism in the world is 2,200 acres in size and one cell wall thick. How is that possible? It is possible because of epigenesis, and these are self-learning membranes. They've learned strategies to be able to constantly combat adversaries and to grow to this huge expanse. Actually, Mark is a friend of mine. He had a small airplane. We flew down there. We couldn't find it. We went back. We looked at scientific literature. We had the long coordinates. We got in a plane again a week later. We flew down there, and we were right on top of it, but we couldn't see it because it was so big. So we spiraled up and up and up and up. We're in a tiny little cloth airplane, and we got to about 15,000 feet. And I've never been in a small airplane, without oxygen. And I told my pilot, Mark, I think I'm going to faint. He said, me too. That's not a good sign. So I said, well, let me get a photograph first. So I took this photograph, which is the best photograph of the largest organism in the world, I think, so far taken. But this is a lesson that we can learn from nature. My good buddy Louis talks about spirals. Well, that's so interesting because these mushrooms also form spirals. Spirals in the petri dish and spirals, in the woods as it gobbles up the trees. Why wouldn't it? It conforms to the basic laws of physics. The mushroom mycelium then is triggered into baby mushroom formation called primordia formation. And at the time of primordia formation, the tips of the mycelium become hypernucleate, nuclear enriched. This is genetic material. Think of it as genetic intelligence. At the tips, at the vanguard of the fungus growing out, it's trying to have the best opportunities to be able to strategize through all the adversarial barriers that it otherwise would encounter. One of my electron micrographs can see the hyperaccumulation partially penetrated through the SEM beam here into the internal nuclei. The primordia then form, then mushrooms then burst. So powerful mushrooms are, they can break through asphalt. This is a shaggy mane mushroom. And we did a series of experiments, and I was working with Battelle Laboratories for a number of years, and breaking down oil using mushrooms. The Department of Transportation in Bellingham, Washington, had a yard where buses and trucks were stored for over 50 years. The Department of Ecology determined in Washington State that this was a toxic waste site, but they gave an exemption temporarily if the Department of Transportation could encourage and prove alternative environmental remediation methods. So there was 4 piles that were selected, absolutely similar. One pile was a control pile. One pile was inoculated with enzymes by a commercial company. Another pile was inoculated with acids, and then our pile was inoculated with mushroom mycelium. The mushroom mycelium absorbs the oil. The mycelium is white, actually becomes black in color. The mycelium then is breaking down the hydrocarbons of the oil and remanufacturing them into fungal carbohydrates. So these are breaking down, molecularly disassembling the carbon into elemental forms, and then they're repackaged as fungal sugars, the carbohydrates that build the cellular structure of these fungi. So we came back 6 weeks later. It was a big contest. The first pile, dead, dark, and stinky, had 20,000 parts per million of polycyclic aromatic hydrocarbons, 2% basically oil, very stinky stuff. You'd have to wash your hands with lava soap just to get the oil off your hands if you touched it. The second pile, dead, dark, and stinky, lifeless. Third pile, the same. And they came to our piles covered with hundreds and hundreds of oyster mushrooms. And notice that the soil changed color. It went from black to white, which is a direct biomarker that the oil and the black was being metabolized and broken down. And some of these mushrooms became huge, a testimonial to the nutrition that they were able to capture from the oil. And so I call these happy mushrooms. They're extraordinarily large. And then, so our pile was teeming with life, and the other piles were dead, dark, and stinky. And then something else happened. The mushrooms would sporulate, they would attract insects, insects would lay eggs, eggs would develop larvae, birds would come in, bring out in seeds, and at the final, and unfortunately the camera didn't work, the final image, this entire pile was a green berm of life. The other piles remained dead, dark, and stinky, and our pile went from 20,000 parts per million of these hydrocarbons to less than 200 in 16 weeks. Then approved by the Department of Ecology as being fully remediated and then allowed for freeway landscaping. Now, the bacterial remediation companies and the phytoremediation companies were concerned. I like to build bridges, not burning them. That's why I love the concept of 4 bridges. This is, in a sense, the concept of 4 bridges right here. And so I said, well, mushrooms are the vanguard species. They break down the toxic barriers to allow the succession of other organisms. And so we're doing phytoremediation. The plants are now growing because they can survive that environment. The spores are attracting insects, and then after the insects are parasitized the mushrooms, then bacteria are growing on the mushrooms. So we have bacterial remediation. So the lesson here is that fungi lower the toxic thresholds that enable the downstream ecosystem to flourish with the biodiversity of life. So you can take an oyster mushroom kit bought from any company. It usually produces 3 or 4 crops or flushes of mushrooms 1 week apart. Then after that, there's not enough nutrition in the mushroom kit to be able to grow more mushrooms. You can take that expired mushroom kit, break it up, take crankcase from your car, pour it on the mycelium, and more mushrooms will grow. Absolutely phenomenal. We analyze the mushrooms. There's no hydrocarbons in the mushrooms whatsoever. But mushrooms hyperaccumulate heavy metals. And so we don't advocate people consuming these mushrooms from toxic waste sites because of the hyperaccumulation of heavy metals. We have disasters. We don't expect these disasters, but they do occur. We know we'll have a lot more in the future. But from the disasters to become opportunities for new solutions. So the Exxon Valdez, Alaska, the Costco Busan, and the BP oil spill all emitted enormous amounts of hydrocarbons and oil into the ocean. I had hundreds of people write me saying, Paul, what would you do? Because they heard about my experiments on land. I've never worked in saltwater before. So I called up several mycologists to ask them, would oyster mushrooms tolerate saltwater? They said, of course not, Paul. I go, how do you know? Did you try? They go, no, but it's obvious. I go, it's not obvious to me. And so I started doing some experiments, and I invented mycobooms. This is hemp socks filled with oyster mushroom mycelium on straw that float. They're only 75% moisture, though of course the water is 100%, so of course it's going to float. The specific gravity is different. And then we invented these mycobooms to corral the oil, and then the emitting enzymes then start breaking down the hydrocarbons, and then something else happened that was totally cool. This is in fresh water, this is in salt water. The alk acid in the mycelium would attract the flies, and the mycobooms then became a huge attractor for insects and fish. And so the fish at dusk would be coming up the mycobooms, going after the insects that were going after the mycelium. We have discovered in the past 2 years a revolutionary method that we're releasing to the public that reduces the cost of remediation with fungi by 100 times. It is the simplest things in life that sometimes are the hardest to see. And I can't believe it took me 30 years to discover this. And this is something we teach in our classes. And then we have a position paper that'll be coming up on the web soon on how to do this. So you don't need equipment. You can just use nature to be able to grow the mycelium. So you can corral the oil slicks together. There's no reason to clean up the mycobooms. They're biodegradable. And we can use the grasses all surrounding in the estuary environments as the material to grow the mycelium. Then we had Chernobyl occur. And Chernobyl created an enormous amount of radioactive fallout. The downstream environment was heavily polluted. And so a group of scientists were very concerned about the consumption of food by the Ukrainians. And so they went out into the forest ecosystem, testing all the wild potential foods and plants in the ecosystem to see if there was any danger for people to consume. Of course, there was great danger. But something amazing occurred from that analysis. They found one mushroom, the hyperaccumulated cesium-137, more than 10,000 times the background levels. So as the radioactive fallout fell onto the forest floor, the mycelium of this mushroom up-channeled the concentrated heavy metals, focused them into a mushroom, which became highly radioactive. But in doing so, decontaminated a wide swath of ground thus allowing the plurality and the biodiversity of the ecosystem to survive, to give rise to the plants that ultimately would feed itself. These skill sets of these species speaks to mycodiversity, the importance of diversity. As we lose species and biodiversity, it's like losing rivets in an airplane. At what point will we go below the threshold where we have catastrophic failure and the ecosystem will unravel? Well, so the Chernobyl became a very exciting experiment. And then after Fukushima, my father was a nuclear scientist. He worked for Babcock and Wilcox. We had many arguments. I protested against nuclear power, made it very interesting dinner conversations. My dad was working for the nuclear power industry, and I was protesting. You can imagine. So I then had hundreds of people write me, what would you do directly after Fukushima? And so here's this mushroom that was discovered in Ukraine, the hyperculeate cesium. And I described within 5 days of the nuclear accident, used to sequester it and create a nuclear forest recovery zone. Don't try to greenwash this. This is a nuclear forest. And this was translated into Japanese, presented to the Fukushima legislature. And in the past 2 weeks, an enormous amount of momentum on this. I have gotten intelligence. I have several friends that are very high up in the U.S. intelligence service, and the Japanese government has been lying to the Japanese people. Up 50 to 100 miles away from Fukushima, there's more than 100 times the safe levels of cesium-137. They're moving 500 truckloads per day. The Fukushima reactor is heating up more now than before. They were venting once a week, now they're venting once a day. What do you think's in the steam? Is continued output of cesium-137. They're bringing in salt water from the ocean to keep the reactor vessels cool, and then they can't discharge that, so they have to store it. So it's a runaway reaction that's now producing xenon light. And so they had cameras there for the public to see, but physicists, universities are going, well, you shouldn't have xenon light. That means the reaction's running away. So then they flooded the vessels with white light. So you couldn't see the xenon light. This is a catastrophe, and their only solution is to scrape the soil all the way down to bedrock or volcanic rock and take it away. Fukushima is a great agricultural and forest area in Japan. It's a breadbasket of Japan. And as they throw away the soil, the breadbasket, is being destroyed as well. So this is a proposal that I made. They asked me, how long will this take? 10 years, 20 years, Paul? I said, no. This will take hundreds of years. 1 generation to pollute, more than 7 generations to clean it up. And so we have to do this in a piecemeal fashion where you harvest the mushrooms out of the radioactive environment, then you incinerate them, and then you capture the heavy metals, or you can use acids to dissolve the fungal tissue, and then the cesium-137 is put on epoxy, and then it's stored ad infinitum. So, but this is the best alternative that the Japanese people have seen in order to preserve their ecosystem and the biodiversity of the environment. If we can remove stepwise fashion the cesium-137, then the ecosystem will recover and be a good food growing area again. So after Chernobyl, a group of scientists were looking at videos inside the containment vessel of Chernobyl. 1 million rads of radioactivity, and they were astonished to see black molds growing all over the walls. On cement, folks, no other nutrition. 1 million rads of radioactivity, and these black molds were going all over the containment vessels. They sampled the molds, and they found something heretofore unknown in science, that fungi use radiation for cellular metabolism in a fashion similar to how plants use light to photosynthesize. So this was a revolutionary discovery. It's unfortunate accidents, but from these accidents, we get new scientific discoveries. I've been involved in Google, and there's a sci-food camp. They bring 140 scientists together, many Nobel laureates, and we all have a brain think tank. I met the leader of the Martian Project. 6 months to go to Mars, 1 year on the planet, 6 months to go back. I said, what's the biggest problem you encounter? He said, by far, food. We have to tow greenhouses in the vacuum of space. I go, how are you going to do that? It's so cold and all the energy. He goes, yeah, it's a huge problem. We can't carry the food. We have to grow it. This discovery here, that the ionizing radiation, both from the reactors on the space vessels and ambient radiation, in space, can be utilized for fungi when we can grow fungal foods. This accident and the subsequent discovery may be pivotal in the ability of humans to colonize outer space. Moreover, as you took your fungal foods with you, you could then terraform other planets because these fungi mineralize the rocks. So you could create the ecosystems and oasis environments that you could expand in the terraforming of other planets. So these disasters, as bad as they are opportunities for coming up with new ideas. This is where Dusty and I live. We have 20 acres on Skookum Inlet. This is the Squaxin Island tribe's land. And this is Totten Inlet and Skookum Inlet. 4 salmon runs a year, super rich in clams and oysters. And I bought this property in 1984, and I bought it, and 1 week later, the sheriff shows up. I thought that was awfully quick. I didn't even have time to do anything illegal. And every landowner on this inlet was served with a summons saying you had to replace your septic system within 2 years or you'd be physically pushed off the property, because the E. coli coming from the uplands was harming the salmon runs, the steelhead, as well as the shellfish, the clams and oysters. I had cows, chickens, and pigs when I bought the property. 2 years later, they came back. 1 year later, they came back and said, Paul, we have an extraordinary thing happening on your property. What did you do? You have more than 100-fold reduction of coliform bacteria. And you have more cows, chickens, and pigs. All of us know they more than double in 1 year. And I had not replaced my septic system. What I'd done is in the swales, I put wood chips. And I inoculated wood chips to mycelium. And the mycelium would hold the wood together, creating that web. And it created a microfiltration membrane and captured the bacteria. This was the dawning of the concept of mycofiltration, a term that I coined. And there's the mycelium, and these giant mushrooms would form. These are up to 5 pounds per specimen, which then would harvest larvae, which I fed the salmon, and then the fish then would eat the larvae that was hosted within the mushrooms. This was astonishingly exciting. Most of the bacteria are trying to kill the mycelium, but the mycelium preselects mutualistic bacteria that helps us host offensive resistant using the antibacterial compounds coming from the friendly bacteria to fight off the bacteria it doesn't want to have. So we have created, and working with John Todd Living Machine, we are now doing experiments. We received an EPA Phase 1 grant for $85,000, which we have successfully completed, proving these concepts in the laboratories at Washington State University. We just now applied for a Phase 2 SBIR to further this technology further to make it open source. So this will be open source technology that we're going to release to the world on using microfiltration for cleaning up water. So Dusty and I decided, well, this is great, but no one has any money. So we decided to pro bono 14 locations in Mason County, Washington. And the water follows the path of least resistance. So the water's going like this. Now, not only is it carrying hydrocarbons, you ever think of where the rubber from your tires go when your tires wear out? It rains, it goes into ditches. Well, E. coli also, so these are choke points. So we put in these bags of mycelium. In this case, coliform bacteria is being tested. This is before up here, and this is about 2 minutes later downstream. We did 10 samples, and generally you can see about a 10-to-1 reduction or greater of coliform bacteria when we put in just 25 of these bags of mycelium in burlap sacks. We worked with the conservation district, created engineering diagrams, and another epiphany came to me that gourmet medicinal mushroom farms should be localized near every community and reinvented as environmental healing arts centers. These are the foundation of the soil ecosphere from which all communities can benefit, not only in helping grow food, but by destroying the toxic waste to make it for a healthier environment. The mycelium produces these enzyme droplets I mentioned, but in these droplets also are antibacterial compounds. And this is a chart, and I have to say I'm married to the most wonderful woman in the world, because I said, honey, we just got our analysis back from this FDA-approved laboratory on the antimicrobial properties of our mushrooms. And I said, but I have bad news. We got a bill for $25,000. I didn't know it was going to be that big. And her first response was, did we get good data? And I went, oh, this is great. And we did get good data. This is a logarithmic scale with E. coli 0157 and MRSA, the methicillin-resistant strain of Staphylococcus aureus, bad bacteria that can gobble you up. And so this is 10 to the first power is 10, 10 to the sixth power is a million, 10 to the eighth power is 100 million colonies in 1 gram of water. We matched it with 1 gram or 1 milliliter of those little droplets you saw, so the 1-to-1 comparison, and 3 species plummeted the amount of E. coli and MRSA 10,000 to 1 in 24 to 48 hours. I took my 10 best guesses, and 3 of them turned out to be very powerful: the oyster mushroom, the birch polypore, and agarikon, which I held above my head. Agarikon has been known as a mushroom for thousands of years to fight consumption, later thought to be known tuberculosis. It is a mushroom that increases the ability to breathe. It has a very nice aromatic effect. If you taste this mushroom, it opens up the lung passages. We tested this mushroom for anti-tubercular properties. Indeed, we found some very potent anti-tubercular properties. Agarikon is the longest living mushroom in the world. It's a perennial mushroom, and it can live up for 100 years, and these are growth rings, and this is the new growth here. And so it has this amazing ability to morph into different forms, and it oftentimes looks like the backside of a very robust lady. And so in ancient times, this mushroom was associated with women pharmacopoeia, with women medicine, and it sometimes can resemble a foot. So with the doctrine of signatures, maybe this is good for fighting foot fungus or foot diseases. It can have the Ganesh form. It can look like an elephant trunk. So it's a changeling. It's a shapeshifter mushroom. It constantly changes its form, so it's hard for a lot of people to identify. I received the National Geographic Award 2 years ago, the Green Innovator Award from National Geographic. They wanted to come out and do a story on agarikon in January. I said, you folks are from New York, you don't understand. There's 10 feet of snow, up in this area, come in July. So my wife and I rented a motor sailor with a Zodiac, and with friends, we went up the inland passage of Desolation Sound in British Columbia. Now, how likely is it we would find agarikon? My professor just found his first one in 40 years. It's about at least 100 to 1, if not 1,000 to 1. So I told the National Geographic guy, I said, well, he said, how likely we will find it? He wanted to find a new finding. And I said, oh, maybe 50-50. I totally did not, and I had a good reason for saying that, because I took up 10 of my friends. We all had high-powered binoculars, and so we went up the inland passage, and we spent so many hours looking at trees. Now, this mushroom grows in living snags. Oftentimes, there's bald eagles. So you see the bald eagles, you look at the mushroom, you look at see if there's a big like wasp nest or hornet's nest on the tree. That's what they look like from a distance. But I figured 10 people, 10 binoculars, going up to Desolation Sound. But you have to realize, it's 5 seconds one tree, no. It's 5 seconds another tree, no. You do that for 2 or 3 hours, you have retina burn, And we weren't doing too well. And a photojournalist from National Geographic said, Paul, I'm on deadline here. We're supposed to find agarikon. We're not doing too well. And I go, yeah, it's okay. It's like fishing. Hang in there. And so we became very distressed but my skipper said, it is a First Peoples site. And we don't know what the pictographs mean anymore. Even the First Peoples, that legend's been lost. But it's very interesting. It's nearby. Why don't we go over and have a break, have a lunch break? And so we motor over to this beautiful place. It has a 30-foot overhang. Remember, there's about 300 to 400 inches of rain. This area ran red with salmon, a perfect, shamanistically perfect place. I believe plants, places, and fungi, mushrooms become shamanistically important because of multiple advantages. The synchronicity of multiple talents that we can benefit from. So we motor over there, and one of our people on our boat goes, there's one. And we found an agarikon at this sacred First Peoples site. It was attached to a branch up here. It fell, it teeter-tottered, and then it grew 2 legs. And the mycelium went back into the mother mycelium of the tree, and they got nutrients, and so no wonder it's called Elixirium ad longum vitam, the elixir of long life. This mushroom doesn't know when to quit. And it takes on these animalistic, that look like 2 legs. So we got really excited, and success. National Geographic was very happy. And Scott Franzbau, who's the director of tuberculosis research, is a good friend of mine. He came with us, and the pictographs are very interesting. And so here's Scott with one of the pictographs, and I have to give credit to my good friend Gujao. Gujao and I are best buddies. He's the president of the Haida people in the Haida Gwaii. Otherwise called the Queen Charlotte Islands, but they don't like to call it that because that's the name of the boat that brought in smallpox. Agarikon is essential to the origination myth of women. And Raven on the canoe traveling in the Sea of Eternity, only Agarikon, fungus man, could help Raven steer on this canoe of eternity to find female genitalia. We must not forget the US and British government were actively involved in bioterrorism against indigenous peoples. I don't have time to go through this, but most of you know the immunological defenses of First Peoples, because we did not have contact with cows, made them extremely susceptible to smallpox. Europeans, 30% of the people would die and exposed to smallpox. First peoples here in North America, 95% death rate. So Gujal told me they had a 10-to-1 kill rate. They killed 10-to-1 the Brits and Americans hiding in the old-growth forest. And the American generals realized and the British generals realized they were losing so many men, they would then sign treaties and give them smallpox-infected blankets. And this is a terrible story, but it's a story that needs to be repeated. Knowing full well they could go into those villages in the spring, and there'd be no resistance because there'd be so much death. So this bioterrorism effort was a military successful strategy by the US and British governments. Gujal did not know how to identify Garikon. Even though it's been resident in this culture, and this Argelic plague is from Charles Edenshaw from 1895, Gujal said we had two flu pandemics, so we had two smallpox pandemics. Each one of them wiped out 90% of our people every 10 or 20 years. We were disconnected from our ancestors because we lost that ancestral knowledge. So Dusty and I felt it was our role to bring back the knowledge of a Garikon back to the Haida and the Haida Gwaii. So we're at this overhang, and I think it's that the pictograph is the same that's in the back of the canoe. We don't know. But we're there, and we see this rock. We look at this rock. Now here's the rock, here's a Garikon, here's the rock, here's a Garikon, here's the rock, here's a Garikon. You get the picture here. I mean, how likely is it we would find a Garikon where pictographs are speaking to the fungus man with the Haida culture? It was 1,000 to 1 to find it, and who knows, 10,000, 100,000 to 1. How likely is it we'd find a rock that looked like it's carved in the shape of a Garikon? And a Garikon was probably resident within those trees far before First Peoples ever discovered them. It's a sacred, shamanistically powerful place. Well, how likely is it we'd find it with a rock like this? I don't know. The numbers are astronomical. How likely is it we'd find it on my birthday? And this is when the photojournalist turned to one of our friends and said, "Does this happen to Paul and Dusty often?" And my friends looked at him deadpan and said, "Yes." And this is where I think intuition and the discovery in science can be led if you are faithful, true to your heart, you respect nature, you respect indigenous knowledge and indigenous peoples, that you will be led to these discoveries. It defies any form of scientific deductive reasoning. So we now today have 45 strains of agarikon in culture, by far the largest library in the world. My lifetime goal is to get 100 of them. This requires us to go out in the old growth forest lots of times. And so now we are going into the old growth forest, and this is the one that I was holding up. This tree it came down, the area was logged. And this is on Cortez Island in British Columbia. And this is a friend of mine, he's a tree arborist and canopy expert, shot his guideline, and Scott Baker then went up this 700-year-old Douglas fir tree and found, and this is where the largest agarikon, the longest living one in the world that we know of, grows. And very interestingly, it does not rot the branch that supports it. It calcifies it. Why would a fungus rot the branch that it wants to support it? It would fall. And so it ended up calcifying, like petrifying the limb. Extraordinarily clever idea. And so it's an epiphyte. Other mosses and other things grow on it, but this is the new growth. And so we have now gotten this strain in culture. We do not pick the mushrooms anymore unless it's going to be logged or there's a fire. And so this is the most recent one. Again, we were led to it. Amazing epiphany. This is in the upper Snoqualmie River watershed. And here's a young agarikon. Now we use GPS coordinates. We make GPS maps of it. And all we do is take a tiny bit of tissue with a stainless steel cork bore, leave the mushroom in the old growth forest, and then we take this amount of tissue, we grow it in culture, and here's an example of 4 strains of agarikon from the same island in the old growth forest. We match them up, and if they create this ridge of resistance, that means their strains are 2 different strains. And so these are different phenotypes. If they come together seamlessly, they're the same strain. So the diversity of strains is really important to us. Now here is an example of it's called a challenge test or a mating test. These are turkey tail cultures. You'll see these in a minute. And this is a turkey tail, 3 strains of turkey tail. These 2 strains, this is the one we used in a breast cancer clinical study I isolated 10 years ago. And then last year, Dusty found this turkey tail on our trail in the woods. And I said, that looks really familiar. So I cultured both of them, matched them up. They grew together seamlessly. We found out that our land has become a fungal library. And so I was able to recapture the strain from nature, and it matched up perfectly, whereas another strain from another area would show the barriers of resistance. This is sort of my life. I do a lot of this. Then the extracellular metabolites, we already know they have antibacterial compounds. We already know they have compounds that break down toxic waste. And then the BioShield program contacted me after 9/11, and the biggest threat that the U.S. Defense Department identified by far is bioterrorism, because people could make this stuff in their kitchen or in their basement laboratory. And Earl Kern is a virologist, an expert in smallpox, and he said that any compounds tested have a 2 or more are active, 10 or more considered extremely active. We started testing our strains, and we're given the green light. The vast majority of 99% of the products we tested were pharmaceuticals. We didn't tell them that we had natural products. We just submitted them in water and ethanol. And we hit the big home run. We ended up having extremely strong activity against orthopoxes, and including smallpox. There is a National Public Radio interview, npr.org. You can listen to it, put in my name, Stamets and smallpox, and you can hear an interview with the Deputy Director of the FDA, the founder of the BioShield program, of the U.S. Defense Department, myself, talking about the importance of this discovery. We had a vetted press release. The vetted press release says more than 200,000 samples tested. We had the most powerful samples ever tested in the BioShield program against pox viruses. In fact, it's more than 2 million. I called up Colonel Huggins, who was my gatekeeper. The vetted press release had to go through the military. I said, well, do you test more than 1 million or less than 1 million? He goes, that's national security, I can't tell you. I go, well, more than 100,000 or less than 100,000? He goes, oh, way more than 100,000. More than 200,000. So it was funny. Somewhere between 200,000 and 1 million was national security, and I don't know where he drew the line. But you can read that press release also. It's vetted by the U.S. Defense Department. And then I was able to make the argument, which the U.S. Defense Department very bought into. Now, some of you may not understand the U.S. Defense Department is designed to prevent destruction of habitats now. They've had this big awakening that destruction of habitats leads to poverty, leads to disease, leads to terrorism. That connection now is very, very well understood. And that's a new thing that the U.S. Defense Department has realized. But I was able to make the argument that therefore, because this mushroom is exclusive in the old-growth forest, we should save the old-growth forest as a matter of national defense. And that argument has gone over extremely well. But smallpox is now in laboratories around the world. Hopefully, it's now extinct in nature. Those of you after 1968, you are not immunized against smallpox, so we're back to square one. But the big virus of concern by far is flu viruses. After Fukushima, or before Fukushima, I was watching 5 bird flu epidemics on the east coast of Japan. And the huge concern that H5N1, the bird flu, will become as communicable as H1N1 as it was 2 years ago, but have a 70 to 80% lethality. If you get bird flu, 70% probability you're going to die from it. But bird flu is not very contagious yet. But after Fukushima, an earthquake, a tsunami, a nuclear meltdown, what do you have? Dead people, chickens, and birds, and pigs floating in a giant petri dish of salt water, exchanging their genetic material. Extinction events occur because of a confluence of characteristics. And we have entered into a period of viral storms. I was recently briefed, and a viral storm is mathematically probable in the next 10 years. It's coming. It's coming soon. It's going to change the very day-to-day activities throughout the entire world. A pandemic is on a near event horizon. And the fact that it hasn't occurred already is rather miraculous. So they started testing our samples that were active against smallpox, against other viruses, since they already had them. And so here's ribavirin, the positive control, pure pharmaceutical. Remember, anything that has 10 or greater is considered to be active. So here's the viruses, H5N1, H3N2, flu B viruses. Here's the positive control. And we gave them our little droplets that you saw from the mycelium and water and ethanol. And here's our results. Now, we gave it to them in 35% ethanol, but we had to dilute it 100 to 1 because these are human cell assays, and alcohol is toxic. So 35% alcohol was diluted 10 to 1 to 3.5, 10 to 1 to 0.35. 100 to 1 dilution of our natural extracts of these old growth, of these fungi that grow in the old growth forest and elsewhere, and we beat the pants off of a pure pharmaceutical. This is the best results that they ever have seen, and this is something that I'm very passionate about. People ask me about patents. You know, I spent hundreds of thousands of dollars of my money on this. I'm up against the big pharmas. Do I want the pharmas to take this from me? No. So I filed patents on this discovery because this is novel, and I am creating, hopefully, a foundation. It'll be a nonprofit to spread this information and knowledge over the world. Viruses know no borders. They don't care about politics. So this is something that we should all be concerned about. And when you die of a viral infection, you die from bacterial pneumonia. It's the subsequent bacterial infection that scars your lungs that proliferates. So to find a mushroom that is dually active against bacteria, like Staph, E. coli, and tuberculosis, and is antiviral, is an amazing package. And when you go around, you go down and do what's called biogated fractionation. You can fractionate and the solvents, you can fractionate to an inactivity. By far, the natural product is a much more useful natural product for building host defensive immunity against viruses and pathogens than trying to find one isolated molecule, which is the pharma school. So viruses can cause cancer. Here are 5 viruses that we know that cause cancer. Lymphoma from the Epstein-Barr virus, or EBV from herpes. Liver cancer from Hep B and C. You know, T-cell leukemia. Cervical cancers cause HPV, and the Kaposi sarcoma, and then Merkel cell carcinoma. So lots of viruses cause cancer. There's a lot more. And if you're immunologically compromised because of bad food, bad health, sedentary lifestyle, and you get a cold, and you get a cut, your immune system is taxed oftentimes beyond its limits. We all have cancer all the time. As your immune system cells are going around, natural killer cells and macrophages and T lymphocytes, they're scavenging them away. So it's really interesting to find a fungus that can be antiviral and activate the immune system and be antibacterial. 73% of all cancer drugs still currently have their origins in natural products. Don't let the pharmas fool you. You know, they are scavenging the natural world constantly, trying to find new medicines. We were funded by NIH for the first breast cancer clinical study in the United States, funded with $2.1 million at Bastyr Medical School and the University of Minnesota Medical School. I was a co-investigator on the grant. We got the $2 million approval from NIH, National Institutes of Health, and my colleague said, well, Paul, we got the grant now, where should we get the mushrooms? I go, please get them from us. There's so many problems in cultivation that people don't understand. And they said, well, you can't be a co-investigator and supplier at the same time. So choose one. So I just said, okay, I'll be a supplier. That meant we were put into a pool of 5 other companies, including 2 from China, that produced these mushroom extracts and mushroom compounds. And so I was in competition. Fortunately, the test results came back. We came out number 1. So objectively, they could say, Paul and Dusty, your mushrooms are the best. We're going to use you. So it's called Tremades versicolor because it's variable in color. You have this mushroom on this land here. It is circumpolar around the world. Wherever trees, you know, cottonwoods, where any trees grow, this mushroom also grows. It's resident within the ecosystem. First peoples, many of you may recognize these mushrooms because they've been put on necklaces and on jewelry. And so the medicine men and women of First Peoples oftentimes would wear the medicines as part of their attire. And so we started working with this mushroom. The breast cancer clinical study has been finalized. It's been published in an international oncology journal. I'll be glad to send you a copy of this clinical study to anybody who wants to receive it. We've now moved on to phase 2. Phase 1 was to determine how it activates the immune system. Prior to radiation, with none of these mushroom capsules being consumed, this is your innate immunity of natural killer cells. 3 grams and 6 grams, you had a direct dose response from taking the mushrooms, your immune system enhanced. When you have radiation, your immune system is compromised. It's depressed substantially.
This is why those of you who have radiation therapy, you're told don't be around sick people. You know, don't get cut. Your immune system's impaired now. We don't want you to die from an infection. So radiation, you know, it helps in destroying the cancer because there's so much collateral damage as well. So at 3 grams and the 6 grams, and then 2 weeks later, you can see a direct dose response curve where the mushrooms increased, and then at 4 weeks, and those of you who like stats, statistically very, very significant. A p-value of 0.0003. And so the significance of this was shown. The breast cancer clinical study has now been published showing immune benefit in combination with chemotherapy and radiation therapy. Now this hit home to me very personally in June of 2009, when my 84-year-old mother then, who was a charismatic Christian who has not seen a doctor since 1968, calls me up and says, Paul, I'm really worried. I need to talk to you about something important, but you're always so busy. I went, oh no, Mom, what is it? She said, my right breast is 5 times the size of my left. I have 6 angry lymph nodes on my right side. Then she started crying and goes, I'm scared. I go, why didn't you tell me sooner? And she said a charismatic Christian scientist came through with a dark field microscope, looked at her blood, they prayed for her and said, you don't have cancer. I believe in the value of prayer. But I said, Mom, you should have seen me sooner. So in June of 2009, we spent most of that month at breast cancer clinics. The Swedish breast cancer clinic, the oncologist said it was the second worst case of breast cancer that she has seen in 20 years of practice. And she should have been seen 2 years earlier. They did MRIs on her across the meridian. It invaded her lymph nodes all over her body. It invaded her sternum, and it went into your liver. Once the cancer gets into your liver and your bone marrow, it's a game over. When you look at the stats, 84-year-old woman with non-ER, non-estrogen responsive breast cancer of this type, the fatality rating is more than 99%. She was put on Herceptin immediately, a great drug. I love Herceptin. And upon the 3rd visit, when the doctor said, ma'am, you should get your life in order, your affairs, you have less than 3 months to live. So we had the circle meeting. I was appointed as executor, and my mother said, I bought a pine casket, the cheapest one I could find. I'm not spending good money on a casket. I'm going to Jesus. I'm going to heaven. So we had the meeting, everyone planned for the end, and the doctor tried to console us by saying, you know, I see ladies in their 40s and 50s, their lives have been stolen from them. Ma'am, you've lived beyond the lifespan of most women. You lived a long life. You should be happy you lived so long. You're too old for a mastectomy. You'll get an infection. Your immune system's depressed. You can't have radiation therapy. You could get an infection. You can take Herceptin. And there's an interesting breast cancer clinical study going over at Bastyr Medical College with this mushroom called turkey tail. You might want to try taking that. And that's why my mother said, my son's supplying that. So she kind of had to hear it from a doctor, you know, before she would take me seriously. My mother started taking 8 turkey tail capsules per day. That was in June of 2009. As of January 2010, and through up to their last analysis this past week, my mother has no detectable tumors whatsoever. I think there's no greater pleasure and payback in life than being able to help rescue a parent from the perils of death. My mother now will be officially cancer-free, that's a medical phrase, at year 5. She's been written up as a best case study in the Journal of Integrative Medicine. My mother, and we now know better what the mode of activity. Not only the natural killer cells are increased, but they decloak the cancer cells. See, cancer grows because it avoids detection by the immune system. And with these fungi, in combination of increasing the natural killer cells, the killer cells can find binding sites using what's called CD8 glycoproteins, are able to lock and load on the skin, the stroma of the tumors, and then it's able to invade inside the tumors and scavenge them away. We've been collecting the best cases. We now have one with lung cancer, advanced stage 4 lung cancer. started taking these mushrooms. The report came back last week. They were analyzed. He was detected a year ago. Was given just a few weeks or months to live. Huge cancerous tumors in his lungs. And now the tumors are hollow. The oncologist said they've never seen anything like this. There's nothing in these sacs. They're just cellular membranes. We have another example of Merkel cell carcinoma. Only 1 out of 10 people in the world have survived this cancer. There is no chemotherapy or radiation therapy for it. It's called Merkel cell carcinoma. It's caused by a polyomavirus. With your genotype, you're likely to get it. This 58-year-old man had lesions all over his body, tumors all over his body. And at Fred Hutch Cancer Center at the University of Washington, when they did a retroactive study, they brought this man in saying, you know, you're not supposed to be alive, and 4 years later, you're alive? What did you do? And he said, I was taking Paul and Dusty's mushrooms. That has now spurred a great thrust, and so we have 3 stellar cases of multiple different types of cancer, which looks like the cancer is being decloaked, and the immune system is not only enhanced, but the immune system then is able to target and invade the cancers. So I leave you today with the strong and documentable belief that nature's complexity has within it many of the solutions that we so need today. Complex problems require complex solutions. In the body intellect of nature, there resides enormous wealth of knowledge that we have not yet tapped into. I'm a strong believer, as you can tell, in evolution. I must say, my mother believes the Earth was created 7,000 years ago. And I said, Mom, look at continental drift. You can measure the drifting of the continents, and you can calculate, you know, going back billions of years. She goes, I don't care. I believe in the Bible. And so that's okay. I respect your opinion. But I strongly believe in evolution, and through the theater of evolution have been hundreds of trillions of experiments that have within resident nature of many of the solutions that we so dearly need. And moreover, I believe that we can accelerate evolution. I'm opposed to GMOs, but I'm not opposed to evolution. We can fast-track the evolutionary epigenetic abilities of these fungi by challenging them with new toxins heretofore not ever seen in nature. But by their adaptability, they can adapt and create strategies that can come up with solutions that can help us today. So I want to just leave. Can I just take five more minutes? Is that okay? This is a new slide. I put it together last night, and this is really important that all of you know. And Rowan, where's Rowan? Are you in the audience? I want to give you credit and thanks because our conversation inspired me to tell you about this. Good. There's my mom. So let's give a shout out for my mom, right? So, and we are facing an epidemic of age. Right now, 20% of the world population is over the age of 65. In 2050, it's going to be that, that's the number of countries going to have populations over the age 65. We have an age bubble hitting, and we have age-related diseases, especially Alzheimer's and other forms of neurological dementia. Women, as they age, are more likely to get Alzheimer's and dementia than men. Women also live longer, so that's a cofactor. But the number of deaths from Alzheimer's-related diseases is soaring, whereas the number of deaths from breast, prostate, and heart disease is going down. Ironic, hard to explain. It's a medical fact. Brain cancer, glioblastoma, and testicular cancer is going way up, probably due to the endocrine disruptors and the pollutants that were in our water. But Alzheimer's is soaring. So we have a huge bubble that's hitting our public health system and our societies, and it's related to neurological dementia caused by Alzheimer's. This mushroom, it may be the first smart mushroom that we know of. This mushroom is called lion's mane, Hericium erinaceus. Dr. Kawagishi gets credit. In 1994, he published a patent, now expired. That's a good thing about patents, they expire. So the researcher invested all of his life, Kawagishi did, in discovering this mushroom that's now in public domain, the knowledge. It contains erinacines. The erinacines are a novel molecule named after the mushroom Hericium erinaceus, and they contain hericinones, another novel molecule that's named after the mushroom. Both of these promote regeneration of myelin. They're called nerve growth factors, causing the regeneration of neurons. And so I've known about this for a very long time, since 1994, And Kawagishi also found this mushroom to be very active against Aspergillus. So it's very interesting. It grows brain neurons, and also fights pathogenic fungi. And so there's 3 reports here, but this is the report that I think it brings it home. They can induce the amyloid plaques that are associated with Alzheimer's by injecting the mice with a peptide. It's a bad thing to do, but this is a mouse experiment. Before they inject it with a peptide that causes Alzheimer's like amyloid plaques, they run the mice through a maze. The mice memorize the maze. You all know about that. You know, they get smart, they know where the food is. They quickly find the food, and they memorize the maze. As they inject it with a peptoid plaque, over a period of about 3 to 4 weeks, the mice lose the ability to navigate. They are suffering from dementia. Another experiment they did. They took the healthy mice, they put it into an arena, and they put a new object, in the middle of the arena. The mice go, oh, new object. They go explore it, really excited about it. They sniff it, they crawl around it, they can measure in time. Doing 10 replicate studies. Wow, these mice all are conforming to the same degree of inquisitiveness, curiosity. When they then injected the mice with the amyloid plaque that causes Alzheimer's, then when they put the old object that was there, when they put a new object in, they wouldn't explore it hardly at all. In fact, they explored it the same amount of time that the old object had been there for weeks, which means virtually no time at all. So they lost the ability of curiosity as dementia increased, as the amyloid plaques increased, as Alzheimer's-like symptoms increased. Then they fed these mice lion's mane mushrooms for 23 days. And the results are as good as to be expected. The Alzheimer-afflicted mice, after receiving the amyloid plaques that developed into full-blown Alzheimer's-like symptoms, eating the mushrooms for 23 days, regained the ability to navigate through the maze. And in the arena, when a new object was put down, they got real excited to explore the object, thus signifying that they had regained curiosity, the dementia had retreated, and upon sacrificing the mice, they can see these amyloid plaques, and they can see the amyloid plaque had disappeared. This is probably the first smart mushroom, and with the biggest tragedy in our military and with our elders and with our wise women and men, they have this body intellect of knowledge all of their life. And at the end of their life, they start losing it. If we can keep our elders, at the top of their game mentally, to pass down the knowledge, the legends, the stories, the language, then we can preserve our culture. So this is the mushroom, which is extremely exciting. I'm writing an article for the Huffington Post once a month. I have 6 of them out. My last article is on lion's mane. So for any physicians or anybody who wants to read these articles and just follow the citations, it's very, very strong. So these mushrooms are powerful for people, places, and planet. They're the very foundation of our ecosystem. They are our proto-ancestors. Looking in the context of evolution, it is clear to me that if we pair with these mushrooms, we respect nature, we respect our ancestors, and we have keen sense of our duties to our descendants, that we'll come out of this in a much better shape. Thank you very much.